We're working on chapter 5. Okay, so in this particular chapter, we're going to be focusing more on controlled studies and expert testimony than anything else. We're not really going to be talking a lot about the analogies. So just keep that in mind as you're reading the chapter. Biggest thing is to, again, pay attention to controlled studies. We're going to break those down and what those will look like. And then what it means to understand who an expert would be in an expert testimony. Okay? Controlled studies, the importance of controlled studies, again, is what the words mean. We're controlling a study. We're using certain reliable methods so that way we can compare results to groups of subjects. Okay, so the idea here is we want to keep a consistent, we're going to use protocol. Protocol is going to be one of the words we're going to use. We use a certain protocol so that if someone tries to duplicate the research in which we did, it should come out with the same results. Oftentimes, if you're looking at research, you want something peer-reviewed. You want something where others have done the same experiment and said, true, what they have found is consistent. It keeps coming out the same for us. We will validate what they are saying. Okay, so if it's not peer-reviewed, I'd usually question it. The science world. It's a whole thing, but whatever. We're just going to try and hone into the simplest parts of understanding what a controlled study is. Again, the methods can be used multiple times for accuracy. It should be able to be replicated with the same results. You're always going to have something that skews the results, but for the most part, you should always get the same results. A controlled study is still inductive reasoning. Remember, we're gathering data, we're gathering research, we're inducting, we're coming up with generalizations based on the data that we've received. Again, it allows the evidence. It's still inductive reasoning. I'm going to read the thing here. It's still inductive reasoning because the evidence allows researchers to draw conclusions. We're able to come to conclusions and generalizations based on the data and the research. So a controlled study is still inductive reasoning. These conclusions are very likely to occur, right? The more you do it, the more you should be able to go, yep, keeps showing up the same thing. And they still answer the same 3 questions. And those 3 questions are, again, characteristic of interest. What is it we want to know? Who's the target population? Who are we trying to do the research on? And then who can I study to find it? What's the sample? Okay, so again, we're coming back. We're going to keep repeating these terms so that we remember what they are. Characteristic of interest is the question. Target population is the who. Sample is the who we're able. Okay, who we're able to sample. Cool. Protocols and subjects. Again, protocols it's the design of the controlled study. It's how you do things, what order you're doing it in. Are you putting on the gloves? Are you, do you have, how are you having subjects enter, exit? I mean, sometimes that does matter depending on the type of research, right? You really have to have a protocol so that every time you do the study, you're having the same results, okay? And if you're not, then you have to ask yourself some questions okay, why, what's skewing this? What's changing the results? Maybe the first one just was a fluke, okay? So the design of the study is the protocol, and if you don't follow the protocol every time you work with one of the subjects, we're going to talk about subjects in a second, then the study won't be reliable, right? So if you don't follow the protocol, when you're working with these subjects, usually patients, people who you're polling, that kind of thing, it's going to skew the results. They're not going to be the same. Subjects are people or animals, right? Usually the people or the animals where you're getting the information from about a target population. And yes, animals can be a target population, depending on how you're doing it. A lot of times they'll use rats because I believe rats have the closest DNA to humans. So that might target population, those rats might still have the results that will apply to humans. Okay, so keep that in mind. Protocol, how in which we do the experiment, subjects, who we're doing the experiment on. Ta-da! Make it easy on you. All right, the ultimate goal when you're doing a controlled study, again, having that protocol, the way in which you're working with the subjects, the whole point is you're trying to weed out the outside factors that could influence the results, right? So we'll talk about double-blind and blind studies in a second, but it's kind of like when you go to do an experiment and you always want to have something that the thing is not done to it, right? So say you are trying to, this is a terrible example, and I'm pretty sure this was found to be inaccurate, but say you're trying to see whether or not negative dialogue to certain things will rot them faster. Okay, just go with me on this one. Okay, yeah, 3 pieces of bread. The first piece of bread's the control, meaning it's going to set aside. We're not going to touch it. We're not going to do anything with it. That's how we're going to base all of our information, is what happens to this thing, right? Because we haven't touched it. Now we're going to do this something else with these other 2 pieces of bread. So one of the pieces of the bread, we're going to say really terrible, mean things to it. And the other piece of bread, we're going to say very nice things to it. And our goal is to see, does terrible negative energy, will that change the bread? Will the bread rot faster than the bread that we say really nice things to? And then we'll compare those pieces to the bread that was never spoken to, which just kind of sat on its own and did its own little bready thing. All right, so that our hypothesis, we'll walk through what the different steps would be. Our hypothesis would be, well, I'm going to say that negative energy will cause that bread to rot faster than the one that I said very nice things to, right? Then we do the experiment. We make sure we keep that control, and everything is controlled, right? Where we're setting the bread in the pieces of, maybe it's under plexiglass. Each one is under plexiglass, right? Each one is in the same environment. They're all the same. That's the control. Everything's the same. The only difference would be what we're doing to these 2 pieces of bread versus this one. Okay. I know that's a really weird thing. I will never forget this experiment. I think they ended up finding out that it was inaccurate, but apparently the bread that was spoken negatively rotted first, or rotted the worst, or something. I don't know. It's not a real thing. But anyway, this is just a little example. Okay, we're trying to weed out any of the outside factors that could influence your results. You want to make sure you have a protocol, right? Make sure everything is the same. You don't want the air to be the thing. You don't want bugs, like a fly lands on this one versus the rest, right? You see they're all encased. Things like that, you really want to be mindful when you're doing experiments, okay? The more you can weed out, the more control you have over the protocol, right? So the more you set everything up where there's not going to be something that's going to change the results, the better. All right, this is why a lot of studies use what we call a control group. That bread that's not touched, that's your control group. All right. Okay, so again, a control group is the group, the thing, the animal, the person that isn't exposed to the variable that your target sample is. So my target sample, it's these 2 pieces of bread, and so, this one again, negative words, positive words, and these are the ones that are having the variable. The variables, my negative words or my positive words. Those are the variables. Those are the things that are changing the outcome of these 2 pieces of bread. Versus this guy who's sitting all by his little self, has not got nothing to do with him, he's just sitting right there. He is my control group. Okay? I hope this is making sense to you. Later on, you're all going to go be like, oh, she's talking about bread. It makes no sense. Okay. So when we talk about a good design for reliable research, I'm sure many of you heard the hypothesis and the thesis and all this stuff. So I'm going to give you what we have in our class that we're going to be using for a good, reliable research. Okay, so first you need to have that question, right? And again, the question could be the characteristic of interest. What's the thing we want to know? What's our question we want that we're asking? We need to know something. So we're going to use the bread. Just go with me on this. Will my negative talk or my positive talk change the outcome of how bread rots, or how, what does it do? Does it true that my negative energy really does create, negative results? That's my question. My hypothesis would be what I'm guessing. I'm going to say, you know what? I think negative talk is going to have a physical reaction to the bread. I think my energy pushing towards it is going to rot the bread faster. But I think if I say positive things, maybe not as much will happen to the bread. Maybe the bread will be the same as the control group. This is my hypothesis. Okay, my sample is, I've got these 3 pieces of bread. Maybe the bread matters. Maybe I need, multiple different types of bread. Maybe it needs to be in different places. I don't know if that's going to make a difference in this particular experiment. That's something you'll want to consider as you're creating your research, right? If you're going to use a sample, what does that sample look like? For me, I'm going to use 3 pieces of the same bread from the same loaf and put it in the same room and with the same temperature. So I'm going to try and keep those things very, very consistent. My sample is going to be from a loaf of bread. And maybe I might try this experiment 3 or 4 different times with different loaves of bread to see. And then the question would be, is it the end pieces that make a difference versus the piece in the middle, right? Is that going to be something? So I really have to consider that as I'm doing this experiment. Maybe my sample is different pieces of the bread in other ways, right? So I have the middle piece breads, and who knows? I'm just saying, think about these things, all right, as you're coming up with your research. And then again, we have our control group. We have that one piece of bread sitting right here. The placebo is going to be something that a lot of times, so the placebo is usually like, it's like a pill, right? Like we say, here's this pill, take it. It's going to be the same pill as everybody else, but it's not, right? It's just a sugar pill. It's just something that we give to see if maybe an animal or a human has a reaction mentally, even though physically they're not getting anything changed. The placebo is just like we call it a sugar pill. It's a non-pill. It doesn't do anything to you. But if you're having a physical reaction, right? So then we could say the person who received the placebo had the same effect as those who received the actual pill. So it could be all mental. So that's why you got to be mindful of a placebo. And then the experimental group are all the people that are within it. Probably not helping that I'm using bread for this, but anyway. All right, and then when you have your independent variable, the thing in which I put like my positive or my negative talk, that's my variable. The pill, that's the variable, right? Whether it's placebo or the other pill, the actual pill given to the patients. Data is all the information that I'm gathering from the results. Like, is the bread rotting? Are the people reacting to the pill? Are they reacting to the placebo, the sugar pill? And then what's my conclusion? What generalization am I going to come, statistical generalization am I going to come to based on all the data that I gathered? Okay, that was a lot. I think you kind of get the general idea. Okay, so again, the question is the characteristic of interest concerning the population. Hypothesis is my speculation on what I think is going to happen. Sample is the member of the target population. Placebo is a pill or a treatment with no actual medical effect. Experimental group is the group of subjects that we're pulling from the sample. Those who are exposed to the special treatment called the independent variable. Again, the thing that I'm giving, whether it's the pill or the positive or negative talk, that's the variable. That's the thing that changes everybody. That's the thing that changes all the subjects at some point, right? Whether they're taking the pill or not taking the pill. Okay. Again, independent variable is the special treatment given to the experiment. Maybe you're being exposed to that independent variable should be the only difference between the control group, that bread, okay, and the experimental group. All right, the breads that I'm telling to. So the only difference is going to be positive or negative talk to the bread or the pill. Okay. Important to keep that in mind because you want to make sure that when you are doing these experiments, there's only one thing. It's that variable, it's that independent variable, that only one thing that's changing it. So that way you can recognize there's not other influences. That's a part of a controlled experiment. Okay. All right, again, the data is going to be the observations that we make, and then the conclusions are the summary that the researcher makes based on the results of the study itself. Okay. All right, we're going to talk about blind studies. There's the blind study, and then there's the double-blind study, and there's two different ones. And please make sure you're using these correctly, use the vocabulary as well. So blind studies are studies in which the subjects, not just the researchers, but the subjects only, are not told whether or not they belong to the control group, which is where they get nothing, or the experimental group in which they get the drug, right? So oftentimes people will be put in, two different groups, the placebo group and then the experimental group. The placebo is the control group, right? The ones that didn't, well, that's a lie, I'm sorry. The control group are the ones that there's no independent variable, nothing happens to them. The experimental group is the one where they did get something given to them, whether it was a pill or whatever the experiment is, okay? That's that difference. So when we're talking about a blind study, the idea is that these subjects have no idea whether or not they are or are not a part of the experiment, right? Again, it's very similar. Like they don't know whether or not the pill they're getting is the placebo or the actual independent variable, okay? So that's the difference between, and that's a blind study. They just don't know. All right, but the researcher knows. Researcher knows exactly who got what pill, exactly got what one thing, like they have an idea. So the blind study is purely for the subjects. Now the double blind is where neither the experimenter or the subjects know, which is the experimental group. So even as a researcher, they wouldn't know. I am actually going to give you a video to watch in just a second, and this hopefully should help you find it, help you get an idea of an example of what it means to have a blind study and a double blind study. And this will be a double blind study experiment.